Emerging research suggests that vitamin B6 may play a protective role in reducing the risk of colorectal cancer, largely through its involvement in DNA synthesis, repair, and methylation, processes that are central to maintaining genomic stability. As the active coenzyme form pyridoxal 5-phosphate (PLP), B6 participates in one-carbon metabolism, where it works synergistically with folate and vitamin B12 to regulate homocysteine levels and facilitate proper DNA replication. When B6 is deficient, these pathways falter, increasing the risk of DNA strand breaks, faulty methylation, and cellular mutations, key mechanisms that can initiate carcinogenesis in rapidly dividing tissues, such as the colon lining. Several large epidemiological studies have reported a significant inverse relationship between plasma PLP levels and colorectal cancer risk. Higher circulating levels of B6 are associated with a lower incidence of both colorectal adenomas, precancerous polyps, and invasive colorectal cancer, independent of folate status. In fact, some studies have shown that individuals with the highest B6 levels have up to a 50% lower risk compared to those with the lowest levels. This protective effect appears strongest in populations with diets low in processed meats and alcohol, both of which are known to increase oxidative stress and DNA damage in the colon, processes that B6 may help counteract. The biological mechanisms behind this are compelling. B6 plays a key role in regulating inflammatory cytokines and reducing oxidative stress, both of which are elevated in colorectal carcinogenesis. It also influences gene expression via methylation of DNA and histones, ensuring that tumor suppressor genes remain active and oncogenes are kept in check. Moreover, PLP-dependent enzymes are involved in the synthesis of nucleotides and in the repair of DNA lesions, an essential defense against the accumulation of mutations in colon epithelial cells. Despite this robust mechanistic support and consistent observational evidence, vitamin B6 is rarely mentioned in cancer prevention guidelines or screening protocols. Most dietary recommendations still focus on fiber, calcium, red meat, and alcohol intake, with little attention to the micronutrient landscape that underlies cellular health. Even folate, which shares many of B6's functions in one-carbon metabolism, receives far more recognition in the context of DNA repair and cancer prevention. This imbalance reflects a broader tendency to spotlight nutrients with more visible or immediate effects, while those with slower, preventive roles like B6 are often overlooked. Complicating matters is the lack of clinical trials directly supplementing B6 for cancer prevention. While ethical and logistical barriers make long-term cancer prevention trials difficult, this gap in randomized data leads to an underestimation of B6's potential in formal recommendations. Nonetheless, the consistent correlation between low B6 and increased cancer risk, especially colorectal, should not be dismissed as coincidental, especially given the known biochemical roles of PLP in maintaining genomic integrity. Another issue is that B6 deficiency is often subclinical and goes undetected. Many individuals, especially the elderly, alcohol users, and those on medications that interfere with B6 metabolism, may have functional deficiency without showing obvious symptoms. In these cases, DNA repair and immune surveillance mechanisms may already be compromised, quietly raising the risk of carcinogenic mutations over time. This makes proactive B6 sufficiency not just a matter of nutritional completeness, but potentially a long-term cancer defense strategy. From a public health perspective, ensuring adequate B6 intake through diet or supplementation could be a low-cost, broadly accessible way to support colon health. Good sources include poultry, fish, bananas, avocados, and whole grains, but the nutrient is sensitive to heat and food processing, making real-world intake often lower than assumed. For those at elevated risk, due to age, diet, stress, or medication use, a moderate supplement of B6, particularly in the active PLP form, may offer an added layer of protection. In conclusion, vitamin B6 holds substantial promise as a modifiable factor in colorectal cancer prevention, thanks to its role in DNA methylation, repair, and inflammation regulation. Yet, it remains largely invisible in mainstream cancer discourse, overshadowed by more headline-grabbing dietary factors. As our understanding of nutrient-gene interactions grows, B6 deserves renewed attention, not just as a metabolic cofactor, but as a potential guardian of genomic stability in one of the most commonly diagnosed cancers worldwide.